Please, Pietro.
Thank you for the introduction. I want to thank the APASL for the very nice invitation. And in particular, I want to thank Satsushi Tanaka. First time I visited Japan was 2006 in a meeting, the liver meeting in Nagasaki. And at that time, I visited Tokyo with Atsushi. We already know each other because we both work with Eric Gers in Davis. Later on, he came in Italy, visiting my institute, and here you can see pictures at my home with Atsushi, and at that time I still have my hair. And later on, I visited my second time in Japan, and we had a good time, great time, spending a family vacation together with Atsushi families and my family. So since then, many times Atsushi came to Italy and for meetings, and our collaboration was really great, and I'm very pleased to be here. Thank you, Atsushi, today. So moving to the topic of my presentation, I skipped the introduction about the pathogenesis of what we know and what we still don't know, because you already heard a lot about. And but clearly we still don't know the reason, the mechanism. The pathogenetic mechanism is still unclear. And all the three autoimmune liver diseases, PBC, PSC, and AIH, has to be considered complex disorders in which environmental triggers and genetic predisposition play a role, with a number of factors here mentioned, and that cause a failure of immune tolerance mechanism with induction of cellular and humoral immune attack, and finally, a pathogenic inflammatory process. So, but today I will focus on sex mainly, and talking about you, not in detail because you already heard a lot about genetics and epigenetics, but mentioning genetics and the role of genetic factor and epigenetic factor related to sex issue in these disorders. But related to gender issue, why focusing on gender? Because for AIH and PBC, we have an extreme female preponderance with, for type 1 autoimmune hepatitis, a ratio 1 to 4 for male to female. For type 2, 1 to 9 as for PBC, with the only exception of PSC, that because of this, can be considered more an immune-related disease more than an autoimmune disease. And this female preponderance is, as you all know, a common feature for autoimmune disorders, as here listed, with a very strong female preponderance in Sjögren, lupus, and thyroid, and you see all the list here mentioned. And for PBC in particular, we have also some exceptional rates. Here in this table, the list of epidemiological data and study performed, and the ratio was consistently very skewed for female, and with some exceptional rates in Estonia, in this Estonian study, for example, 1 to 22. With some exception, recently, starting from our own study, recently published, we started to debate about the ratio, male-to-male ratio in PBC. And as you can see here, in this study, we analyzed the administrative data from a northern Italian region, Lombardia, with 10 million inhabitants. And so going through the administrative data of our national health system, we were able to identify, by using the code, the specific code for the disease, almost 3,000 patients. And in this case, the male-to-female ratio was 1:2, so more male patient than expected. And the same exercise was repeated in the Denmark registry, using the Denmark registry, again, using and analyzing the administrative data. So Danish people are 5.5 million. And also there, we identified 700 patients with a female-to-male ratio 1:4. So after this first report, other papers came, studied, and papers were published reporting similar data. So the questions is we are missing some male patient with PBC, and so discussion is still open, and we are looking for more data in different geographical regions and more, and actually an appropriate explanation. So, but coming to the female preponderance in autoimmune liver diseases, why? So let's start with some observations. So female preponderance is a consistent feature of autoimmune disease, so as I already said. So, and then a considerable number of sex and immune-related genes are located in X chromosome. And major disorders of X chromosome, such as Turner syndrome, that is characterized by the lack of an entire one X chromosome, or X-linked immune deficiencies, are associated with many autoimmune features and autoimmune diseases, and as well as cholestasis. So, and then move to some, so we don't have an answer, but we have several hypotheses. One was related to the role of sex hormones. So then, and let's consider that immune cells express receptors for both estrogen and progesterone. And so that their activity can modulate by sex hormones on numbers of immune-related cells, actually all the immune-related cells, B cells, T cells, monocytes, and so on. So, and maybe it's too simplistic, but estrogen has mainly a stimulatory effect on the immune system, while progesterone does have an inhibitory profile. And even considering this, looking at the available data, today I think it's fair to say that the imbalance of sex hormones cannot explain the female preponderance in autoimmunity and in autoimmune liver diseases. Another hypothesis, the fetal microchimerism, that is the mutual exchange of cells with a different genetic background between the mother and the offspring. So several papers and studies show that fetal cells can be found in the liver, can persist in the liver, in the blood, for many years after delivery. And so it was hypothesized that might trigger autoimmunity. But we have also some data in PBC patients and other autoimmune liver diseases, but today we can say that works are in progress on this. And it's not the real, I think it's not, will not be the answer for the female preponderance. And then another hypothesis, the role of genetics, and as you will hear from me now, also epigenetic factors associated with X chromosome. So, and we have two possible approaches on this hypothesis. One is studying the monosomy X, and then the GWAS, or actually better, the XWAS approach, sorry. So we started with studying several years ago the monosomy X in PBC. The method we applied was by FISH, and here you can see 5 nuclei from a female PBC patient, where you can see the green or pink spots are the control chromosome, chromosome 15. Each nuclei has 2 spots, but looking at the green spot, that it means the presence of X chromosome, you see 3 nuclei, 1, 2, 3 nuclei with only 1 green spot. It means only 1 X chromosome, that is monosomy X. So by counting at least 500 nuclei from each subject, patients and controls, we found an increased monosomy X level in female with PBC compared to female with similar age. And this increase, monosomy X, was present in all the major immune cells we evaluated, CD3, lymphocyte, monocyte, and so on, as you can see. And we also able to find the same defect, a major genetic defect in X chromosome in other autoimmune diseases, such as autoimmune thyroiditis and systemic sclerosis. And finally, we also more recently found also a defect in the Y chromosome. So an increased Y chromosome loss in male with PBC compared to healthy male. Again, with compared to male, healthy male in different ages. And so considering all this data, so an enhanced X-monosomy in females and preferentially X chromosome lost in female, an increased Y chromosome loss possibly due to an instability of sex chromosome in this disease, we came to a unifying hypothesis. That is that in this disease, patient developed a sort of haploinsufficiency that unmasked some important genes, susceptibility genes. So regarding monosomy X, I think all we could do was done, except that we need some independent confirmation. So just if you, some of you is interested in this, we need a confirmation. And then I move to the X chromosome XY association study. And I'm going very quick because you already heard a lot about the GWAS data in PBC, and you already heard that here 3 Manhattan plots from 3 different GWAS studies, and you already heard that HLA was the strongest association found in all the GWAS studies performed in PBC, but was also the main risk locus found to be associated in PSC and AIH. But again, you already heard that a number of other non-HLA association was found, and here the growing number of loci found associated and loci coding for genes related to the immune system, all of them. And here some consideration about the sharing of association with other diseases, autoimmune diseases. Here, I think you cannot see very well, but all of these cycles is related to a different autoimmune disease. And here, all the chromosomes with 22 chromosomes and X and Y. And what we can say here, that the maximum overlaps correspond to HLA region for not only the 3 autoimmune liver diseases, but autoimmune diseases in general. And what about X and Y chromosome? So we have a lack of association, not only in PBC and autoimmune liver diseases, but in any other autoimmune diseases. Why? So X chromosome is quite a huge chromosome compared to the other chromosomes. It contains more than 800 protein coding genes, and so on and so on. So it's very strange. It might be, it's very strange not to have any association, gene association in these chromosomes. And for this reason, years ago, we moved to epigenetics, thinking then genetic aspect related to X chromosome were not were not important. But then we realized that possible reason for this lack of association was methodological, because GWAS did not analyze systematically sex chromosome, and we lacked for years of an appropriate bioinformatics tools to analyze sex chromosome. And so we decided to start this project and to analyze the XWAS, to have an XWAS approach, not different from GWAS approach. And so we started collecting the GWAS data from all over the world, as you can see, with also contribution for Japan and China, Asian GWAS data. We applied for a novel bioinformatics tools and approach. I'm not going in detail, but the results were quite interesting. We were able to find clear and strong association with some loci and genes in X chromosome. And here the list, and as you can see, all of them were, the stronger association was in the Asian subset of patients. But considering the hereditability of autoimmune liver diseases, we can explain only 20–30% maximum. And so we are missing something. And here the list of what can explain the remaining hereditability. But I want talking about the female preponderance now, moving to epigenetics. And you already heard a lot by Professor Liu about, so I'm going very quickly. So the current definition is a stably heritable phenotype resulting from changes in the chromosome without alteration of DNA sequencing. And here the principal player, mainly methylation, but others. And so when we have problems in methylation and other players, we might have a disease. We might have consequence and different phenotypes and disease. And if we see the literature, if we look at the literature related to autoimmune liver diseases and epigenetics, we see that is a field now growing, but still very small, and most of the data are related to PBC, and mainly on DNA methylation is one of the most important players in epigenetic field. And with the methylation, that means the methylation, more we have the promoter genes methylated and less we have gene expression. So we have a number of data related to epigenetics in PBC nowadays, some data exploring the whole genome methylation status, or a single chromosome methylation status, or some data with specific genes, a single gene level, candidate gene methylation changes. And I want just to elude two papers because related to methylation status related to sex chromosome, X chromosome. One was a paper on X chromosome, the whole genome, whole methylation status of the entire X chromosome, another related to a candidate X-linked genes methylation. So the first, we performed this study, extensive X chromosome methylation profile, in the main immune-related cells, CD4, CD8, CD14, in a large number of PBCN controls, and here the most striking results, that are the methylation of this very important immune receptor, immune-related receptor, CXCR3, that was demethylated in all the three immune cells evaluated in the study. We also found some hypermethylated genes, but different in different cells. So studies on the role of CXCR3 are undergoing now, of course. And then the other example is a different approach, so a candidate X-linked genes. So we evaluated CD40 ligand gene. CD40 ligand, the crosstalk between CD40 and CD40 ligand has an important role is an immune system, immune tolerance, and it's very important that the genetic defect in this gene lead to an X-linked immune deficiency, the hyper-IgM syndrome. And you remember, all of you knows that PBC patient has high levels of IgM, and the reason was still unknown, or was unknown. So we performed the study studying CD40, CD4, and CD8 T cells. We extracted DNA, RNA in patient and healthy controls and disease controls, and we found a reduced gene promoter methylation of at the promoter of the CD40 ligand gene, as you can see here, the red bars compared to the other one. And we also found a differential gene expression of CD40 ligand, more expressed in PBC compared to healthy control, and this, of course, correlate with methylation. But what was very important was the finding that the highest IgM levels correlate with the lower CD40 ligand promoter methylation. So we believe this is the explanation for IgM higher level in PBC. And so I hope I convinced you that the female predominance in PBC now, but we believe also for other autoimmune disease and AIH is due to genetics and epigenetic defect in Gen X defect. And clearly we are still on top of an iceberg. So we clearly understood that there is something wrong, but we need more and more studies for sure to solve these important questions. Because to understand, in my opinion, to understand the reason for the female preponderance will give us the answer also to the reason of the disease. And I want to thank you for the attention and my group for the support. Thank you.
Professor Pietro, thank you for your wonderful and comprehensive lecture. So it's open for discussion. Any question from audience? Okay. In your abstract, you mentioned the animal model overexpressed of interferon gamma have a clear female predominance. Can you
speculate?
Yeah.
Yeah, actually, that was a very, very important finding because thanks to this model, we might go more in deep to understand the reason for female preponderance in this disease, but not only in this disease. So we are currently working on that. I cannot say more now, but it was a striking and finding and very important one. And of course, supporting the role of interferon gamma in the development of this disease.
Thank you.
So, are you doing any more work on identical twins, or has that stopped?
It's difficult. Stop because it's a rare disease and will be very difficult to find a larger number of pairs, the twin pairs. And so that was the main reason. The tools, in theory, would be fantastic to go on and trying to understand all of these issues. It's the role of genetics, epigenetics. But as you know, when you were able to collect 15, 20 pairs, twin pairs, that was exceptional. And now, I don't know, maybe we should
need to go back and make a big effort again,
yeah,
considering what's happened.
Yeah.
Yeah.
Well, thank you very much for introducing our very old pictures. Anyway, our recent epidemiological study in Japan also indicated the great change in the male-female ratio. So we used to have about 1 to 9. About 20 years ago, but last year we found that the ratio was 1.4, just like your data in your Lombardy. So I think that would be might be a clue to explore the reason why the female predominance is observed in PBC. So how do you think about it? How do you explain the change regarding your hypothesis?
I have only one very easy possible answer, and is that most of the patient with PBC has a mild and asymptomatic disease, as we all know. And so when we make the diagnosis in male patient with a very mild disease, and we ask them just come regularly every 6 months, yearly, for a visit, do controls, most of them, they don't do.
Okay. Thank you.
Yeah, nice talk. I have an interest about the X-monosomy. So, did you have any ideas that these cells work normally, or did you check the biological function of the X-monosomy cells?
Yeah, we checked in the X-monosomy, actually the presence of X-monosomy by fish fluorescence inside the disease. Yeah, and was very marked, the difference between monosomy X cells among T cells and monosomy X in T cells from healthy women.
Okay, so do these cells work normally? Normal?
I don't know.
Okay.
I don't know. Yeah.
Okay. Thank you again for your nice talk. Okay.
Next talk is by Dr.